In scientific research, an experimental system is the physical, technical, and procedural basis for an experiment or series of experiments. Scientists, particularly laboratory biologists, and historians and philosophers of biology have pointed to the development and spread of successful experimental systems, such as those based on popular model organism or scientific apparatus, as key elements in the history of science, particularly since the early 20th century. The choice of an appropriate experimental system is often seen as critical for a scientist's long-term success, as experimental systems can be very productive for some kinds of questions and less productive for others, acquiring a sort of momentum that takes research in unpredicted directions. A successful experimental system must be stable and reproducible enough for scientists to make sense of the system's behavior, but variable and unpredictable enough that it can produce useful results. In many cases, a well-understood experimental system can be black-boxed as a standard technique, which can then be utilized as a component of other experimental systems. Reinberger divides experimental systems into two parts, the part under investigation, epistemic things, and the well-understood part that provides a stable context for experimentation, technical objects. The development of experimental systems in biology often requires the domestication of a particular organism for the laboratory environment, including the creation of relatively homogeneous lines or strains and the tailoring of conditions to highlight the variable aspects that scientists are interested in. Scientific technologies, similarly, often require the development of a full experimental system to go from a viable concept to a technique that works in practice on a usefully consistent basis.